Hi everyone, my name is Rory Chisholm and I'm part of the Analytical Chemistry team here at Dalhousie University. One of our main teaching philosophies in regards to teaching analytical chemistry in the lab is learning through active participation. And what I mean by that is where students get to interact with each other, problem solve, troubleshoot, collect live data, and make mistakes and learn from those mistakes. And so as Dalhousie had to close down our chemistry labs due to COVID-19, we had to reevaluate how we deliver this laboratory to our students, yet still maintain student engagement. As such, we developed a very intense schedule for our 13-week term for our introductory analytical chemistry students. 12 of those weeks involved the students meeting in small groups, 3 to 5, with a TA. In these small remote lab meetings, as I called them, the students would share and discuss their work for pre-lab questions with each other. Also, I'd like to point out 6 of the weeks during the term, students collected data and did experiments at home without any kits being sent out. And today, I'd like to focus on 3 of these weeks in which students used a free cell phone app that they downloaded onto their cell phones to be able to better understand absorption measurements and relate these absorption measurements to Beer's law. We chose to introduce the use of a cell phone app to collect data at home very early in the term, right within Experiment 1. Traditionally, Experiment 1 is an introductory lab easing the students into our analytical lab. In this, students would traditionally collect data and compare 2 different data sets using a variety of different analytical statistical analysis. We did this by providing the links for one of the 2 LuxMeter apps for our cell phones. So you could either download an Apple LuxMeter app or an Android LuxMeter app. Following this, we provided online tutorials for students how to actually use these LuxMeter apps on their cell phones at home. Students were provided tutorials on how to optimize and align their cell phone LuxMeter sensor on their cell phone with either a video that was provided online or images. Experiment 1 consisted of students using their LuxMeter app to be able to compare the intensity from 2 different flames associated with calcium emission. The 2 different flames were generated from slightly different calcium concentration solutions that to the eye seem basically the same. Using the large data pools that the students collected for the 2 different flames, the students did a variety of different statistical techniques that we use in analytical chemistry. Following this initial activity of students using a cell phone at home to measure light intensity through the computer, Experiment 3 consisted of, again using the cell phone app, but then to be able to convert these light intensity measurements to absorption. To do this, we captured images of iron orthophenolate solutions ranging from 0 to 10 ppm. You would then measure the intensity of your blank solution on our far left, and this would be our I naught. By measuring the intensity of the ever darkening solutions, you could then convert those into absorption. Once you have the absorption for all your various solutions, you could plot this as a calibration curve. And what you find is when comparing our cell phone app to a commercially available UV vis that has been selected for the wavelength of maximum absorption, you find that the cell phone app actually measures a 10-fold decrease in sensitivity. Students then measured the intensity of an extract solution and then back calculated the absorption for this. By then applying the calibration curve and Beer's law, they could calculate the concentration in their extraction solution and then back calculate the amount of iron located within a multivitamin tablet. Overall, out of a class of 132 students, approximately 78% of the students chose to use their own cell phone app and data they collected on their own, even though data was available for them to download and process to complete the lab report. And what that demonstrates to me is students want to be involved in data collection despite being remote and scattered all over the world. And with that, none of this would have been possible without the support of our teaching assistants. So I had 7 very talented graduate student teaching assistants working with me. One I'd like to particularly point out, Jessica Nickerson. She was heavily involved in Experiment 3 development. I'd also like to point out various funding agencies that supported this classroom development.